an area that's quite hot right now with lots of clinical trial activity going on. Oh boy. Oh, disclosures relative to PARP. So we've all seen this slide many, many times. We now know that about a quarter of metastatic castration-resistant prostate cancer tumors harbor some form of DNA repair alteration. As you can see, there's a broad, there's about 6 or 7 genes that showed up on this Robinson paper. The reality is there are many more genes that are associated with DNA repair. And as I'm getting more and more genomic testing results back, what I'm seeing is that in the reports, in the dictated reports that we get, many, many genes that are loosely associated with DNA repair are listed, some that I've never heard of before and aren't on this relatively short list from the Robinson paper. However, we don't know the degree to which they are all associated with DNA repair, and I think for the time being, we need to really focus our efforts on BRCA1, BRCA2, and possibly ATM as we move forward with clinical trial development. So I think this is a slide that we maybe could have shown as we were talking with Oliver here in the last segment, because there are many types of DNA repair. DNA repair is not one monolithic entity. PARP is actually involved in single-strand break repair, which is different, for example, from what ATM and DNA PK do, which is double-strand non-homologous end-joining repair. And so, you know, this slide is highly theoretical, linking the different types of DNA damage to different mechanisms of DNA repair to different damaging agents. And so, really, what we're thinking about here is inhibiting the DNA repair process in this sort of synthetic lethality idea, which is to say that if a patient has a tumor that already has a defect in DNA repair, it has a compensatory mechanism. That's what I think of PARP as, and by inhibiting PARP, we lead to the synthetic lethality. The tumor goes lethal because it cannot synthesize new DNA, because it now has 2 hits on its DNA repair. PARP is important in DNA repair, but I think it is also just important to say that it's important in all of us in the normal cells and HRD cancer cells. Why would it be any different? So the idea here is that PARP gets trapped. It goes onto DNA to repair double-strand breaks, et cetera, and if PARP is inhibited in a normal cell, there's a homologous recombination. There's a second or a third. There's an other mechanism of DNA repair. And you can imagine evolutionarily that it would make sense to have a wide variety of DNA repair mechanisms. Otherwise, we wouldn't have evolved and survived the way we did with all the damage that UV light and other things do to our DNA. But again, in the cancer cell situation, it's not able to compensate for that, leading to DNA accumulation and cell death. So that's kind of the idea. TOPARP was the study that really caught everybody's interest. You know, we had known for years that there was this sort of BRCA story in prostate cancer, and it's funny, I had William O. had kind of coined this concept many years ago, and I had a protocol sitting on my laptop for many years, BRCA protocol that I never worked on, that William O. had sent, and it was sort of languishing, and then all of a sudden the TOPARP data came out, and I said, well, there goes that, and this was a study in which they took unselected CRPC patients and just really wanted to look at patients with response to olaparib, and they also had sort of this process by which patients who had a high response rate, they would do a randomized study in unselected patients. That was the way they designed their phase 2. But if they had an intermediate response rate, they would look at selected patients and consider making this a biomarker selected drug. And that's really what emerged, was that they didn't have a high response rate, but they did when you think about selection. So they treated an initial 30 patients, and they had 20 patients only who had this sort of intermediate, 20 patients in the biomarker selected group. So they had a total of 50 patients to evaluate for response to olaparib in biomarker selection versus no biomarker selection. And these are the results, and they're quite impressive, and that's why it went into the New England Journal of Medicine. In the patients in whom they were biomarker selected because of the presence of a DNA repair alteration, the median PFS to olaparib and CRPC, and many of these are post-chemotherapy patients, was 9.8 months, where it was virtually 3 months, or a little bit under 3 months in the unselected group. And in the patients who had the biomarker positive group, the overall survival was 14 months, which actually compares very favorably, for example, to the 301 abiraterone and enzalutamide data in the post-chemotherapy setting. But the most important thing is clearly biomarker selection was important in this setting, and I think that was the take-home message. So that's take-home message number one. Take-home message number two is that PARP enzymes actually do a lot of different things besides DNA repair, and there may be a window of opportunity here in prostate cancer insofar as we might be able to target the transcriptional modulation aspects of PARP1. PARP1 collaborates with the AR and ERG binding and supports their transcriptional function. And so it does make, there's actually a sort of a better story by saying that AR helps enable DNA repair. It's not so much that PARP helps AR, they're cooperative in a way, but as we think about castration-resistant prostate cancer, it is important to think that, or to know that castration-resistant prostate cancer have increased PARP activity even in the setting of even in a non-BRCA setting. So that is potentially something that could be exploited, and we keep talking about how we should be exploiting this non-DNA repair pathway, but I just got done showing you clinical trial data where we didn't see much activity of PARP inhibitors in non-selected patients. So we've got to be careful about how much enthusiasm we put around this concept. Nevertheless, there are some interesting data that come out, and maybe many of you have seen this. This is a study that was led by Maha Hussein when she was at Michigan, and Felix Feng did the genomics on this, and a wide variety of people participated. But basically what this trial showed was that patients who had deficient DNA repair who were treated on, this trial was abiraterone versus abiraterone plus veliparib. But what they showed is that patients who were on abiraterone alone, who had BRCA mutations, actually had a benefit over patients who did not have BRCA mutations. So again, that suggests that inhibiting AR, or androgens, in a DNA repair defective setting may be sufficient to promote some clinical benefit in some patients. Veliparib is not a particularly potent PARP inhibitor compared to some of the others, and that's one of the things that is continuing to be one of the stories that's continuing to be sort of worked out. Olaparib is more potent. There are a number of studies. This is a table I took. I actually don't have the date from when this was in terms of where they are, but there are many studies that are looking at olaparib in prostate cancer, in castration-resistant prostate cancer, and there are 3 studies going on. Two phase 3 studies of PARP inhibitors that I know of in CRPC, and I might be missing one. The PROfound study is being led by Johan de Bono, that's shown on the bottom. That's taking patients with prior abiraterone or enzalutamide with a BRCA1, BRCA2, or ATM mutation, and randomizing them to olaparib or physician's choice. The Triton3 study, of which I am the PI, is very similar. It's a post-Abi-Enza, BRCA1, BRCA2, ATM, randomizing 2-to-1 to rucaparib versus standard of care with intense crossover. So everybody will get a PARP inhibitor. And then there's I believe there's another study underway that Maha is leading, looking at CRPC patients, looking at olaparib versus abiraterone versus olaparib plus abiraterone, which I believe is up and running in BRCA1, BRCA2, and ATM patients. I think it's a little problematic, all the lumping we're doing with BRCA1, BRCA2, and ATM, and I think that may be something that we'll want to look closely at down the road, because these have different associations with DNA repair and different associations with PARP. If you caught it, and I went through it very fast, but in that initial cartoon that I showed you, PARP and ATM are sort of in different columns in terms of their DNA repair, what they function in DNA repair. So not all DNA repair alterations are the same. We have, through collaborating with Alex Wyatt and our West Coast Dream Team, been able to uncover some potential mechanisms of DNA repair potential mechanisms of resistance to PARP inhibitors, and I think it's really very fascinating, so I thought I'd bring it up. So what we have in, for example, a BRCA2 mutation is you have a truncated, you have a mutant protein which is unable to function, and through clonal selection or through selection pressure, there's essentially a second mutation that can evolve in BRCA2 that can essentially result in a truncated protein, but one where the BRCA function or the BRCA function is restored. So you get a mutation that undoes a prior mutation, and so therefore they're called reversion mutations. And This is something. This is something that that has been observed in the small number of patients who have been sequenced post-PARP inhibitors at the point of resistance. Almost all of them have these reversion mutations. Some patients have many reversion mutations. And so at UCSF, we had a protocol where we're collecting DNA on these patients as they develop resistance to PARP inhibitors, and the PCF is funding that work. You know, one of the challenges with PARP inhibitor development also is getting to the tissue. I just wanted to highlight Kim Chi's work. It's from the Annals from Cancer Discovery earlier this year. He presented at ASCO. They looked at circulating tumor DNA and showed that BRCA2 mutation found in circulating tumor DNA conferred a much worse prognosis and basic resistance to abiraterone. And so this data flies in the face of Maha Hussein's data, which said that BRCA2 mutations lead to sensitivity to abiraterone. So that's an interesting sort of little story debate that's happening. We'll have to see what happens there. And this is just further data showing that they can use the ctDNA, which was highly predictive, looking at a number of factors. And I appreciate that the slides are kind of small. I apologize for that. But anyway, the point of this slide is to say that cell-free DNA, I think, is the wave of the future in terms of analyzing this with a liquid biopsy. This is data that I won't go into with reversion mutations. My 10 minutes went by really fast. Anyway, so what we don't know with regard to the PARP inhibitors, and I looking forward to conversation, is what is the best setting? You know, if we know that somebody has a DNA repair defect, should we put them on a PARP inhibitor right away? And this happens in my clinic. You know, the fellows are all excited, and they'll see a patient with high-grade, high-risk, newly diagnosed metastatic disease, and they'll want to sequence them right away. And I say, well, that sequencing data could be great, but we may not need that for 2 years or 3 years. And we have no data that early PARP inhibition or other targeting of DNA repair alterations is efficacious. And so those are kinds of things that we want to look at. PFS endpoint I won't go into. In combination, we thought when these data first came out, oh great, we can take these PARP inhibitors, we can give platinum because platinum damages DNA, will have this sort of synergistic effect. It turns out that every study where platinum has been combined with a PARP inhibitor has been very difficult from a marrow suppression perspective. So I don't see that moving forward as a treatment modality unless we can work seriously on reducing the dose of platinum or alternating courses or things like that. So lots of things to think about. I think the major issue that we face with regards to PARP inhibitors in prostate cancer is that these drugs are already FDA approved for ovarian cancer, and that patients can get their hands on them. And we have about 4 or 5 or 6 clinical trials trying to accrue. And patients, when they find out they have this DNA repair alteration, and when we say we're going to sequence them to find out if they have a DNA repair alteration, they don't want to be randomized to some control arm. And so I think this is a real challenge. It's really hard work getting patients accrued to these trials in the U.S. In Europe and in other countries, they don't have that sort of a liberal access to these therapies. And so that's probably Europe is probably going to, and Australia are probably going to carry the day in terms of accrual to these studies. So with that, I will stop and take any questions and hope we have a good conversation about PARP inhibitors.
You know, Chuck, do you think platinum's a poor man's PARP?
I think so. Platinum is yes. Platinum has some advantages over PARP inhibition. I mean, PARP inhibition targets one process, one very specific mechanism of DNA repair, whereas platinum damages all DNA. So theoretically, a patient with a CDK12 mutation who may not have PARP activation could be sensitive to platinum, but not to PARP inhibition. So that's a reasonable thing to think about. The best PARP inhibitor responses that I've seen in patients who've had prior platinum, but who were not treated to platinum resistance. The DNA repair mutant platinum resistant patients also have reversion mutations, and that's been shown in ovarian cancer. So I think a PARP versus a platinum study would be interesting to do.
I got it. When you look at PARPs, I mean, all PARPs are definitely not the same.
Yeah.
And there's both the enzymatic inhibition, but then there's the PARP trapping.
Yeah.
You know, that some of the ones say that may be more important.
Yeah.
I mean, is that being looked at more, especially in prostate cancer, to see, again, one of these going to be better, one is going to combine better with other DNA damaging agents? Anything you know about kind of looking at those two mechanisms on how you may want to look at these PARPs.
Yeah, my personal bias on that question is those are interesting in vitro observations. They were interesting observations for the respective companies to make as they proceeded into the clinic and to try to convince us to be involved with their particular agent, if that was the case. But we'll see what happens as it bears out clinically. It reminds me a little bit of abiraterone or Teranel and galetterone and enzalutamide, all of which sort of hit the clinic at the same time in around 2008. And every one company was saying, well, we don't need steroids, and other ones, we target. And it turns out that it just took all the clinical trials to sort out which were the most effective drugs in that setting. So I just don't think we can use that knowledge anymore. They're all in the clinic. We'll see what happens.
So should it be potentially a cooperative group trial? We have a relatively cheap generic agent and a very expensive oral agent. And, to be blunt, I've kind of been shocked at the price of the PARP inhibitors. Should we be doing a trial or promoting a trial, platinum versus PARP? Do you think it's doable?
Yeah, so I lead the Advanced Prostate Cancer Cadre in the Alliance, and my job is to come up with trials for advanced prostate cancer, and we've talked about this a lot. So first of all, we think in the cooperative groups, we can't compete for the PARP inhibitor development, story with the companies, right?
Agreed.
But we could do the platinum piece. And so what we would have to do at this point, though, would be to get a company to give us their drug to compare it to platinum, right? So no company's going to give us their expensive PARP inhibitor and say, here, go compare it to a generic, easy-to-administer chemotherapy. And so that's one point. And the other point is we had talked briefly about doing a NOVA-type trial. The NOVA trial was in ovarian cancer, and that's a very platinum-sensitive disease where everybody responds to platinum. But what they did is they gave platinum, and then if you responded, they gave you PARP maintenance, and that was a profoundly positive study for RUCAP. That was niraparib, but the same paradigm has been done with other drugs. And I propose that we think about that in prostate cancer, but the reality is we would have to get enough patients to analyze PARP maintenance versus no PARP maintenance. We'd have to get platinum responders. We'd have to, get like 2,000 people to identify 500 platinum responders if we're lucky. Then do the randomization and 250 per arm. So it's just like a heavy lift in terms of selection of patients.
So I think Bruce Montgomery's VA study will talk about the platinum pretty well, and then that'll be the gold standard.
Yeah.
Okay.
Chuck, once you get it approved, though, once you get an agent approved, you could do a randomized trial at that point.
Yeah.
You wouldn't need to have a pharma company sponsor you.
Yeah.
And I don't think you're that far off from getting.
CTEP would have trouble on its drug budget.
Really.
Yeah.
If it's not provided, then somebody has to pay.
But the other thing you could do is do a thing where you treat with platinum briefly, you treat with a PARP, you treat with platinum, do those kinds of, there are all kinds of those types of questions that could come up. But I still think there's so many questions in this field. I used to think it seemed pretty tight. And the more I've started getting patients sequenced and I'm trying to treat ATM mutations and BRCA1 and BRCA2 and the various different, mutations, it's a very muddy picture because it's not like it's one biomarker of one biological entity. It's a very diverse biological thing.
Chuck, sorry, Julia, I don't mean to get us off schedule. Just one quick question. So in ovarian, there really is this concept of BRCA-ness, which you apparently had on your desk for a while as a clinical trial proposal. But for prostate, what do you think the difference is between prostate and ovarian in that BRCA-ness? Or have we just not, are we too early in our understanding of this entity in prostate cancer that we haven't defined BRCA-ness? It doesn't seem to be as present in my clinic, I would say, as it clearly is in ovarian. So in that trial that you mentioned, those patients on maintenance, even when they didn't have DNA repair defects, there was a huge response.
That's the point. Yeah, that's the key. I think is that ovarian cancer is a majority platinum sensitive disease, whereas prostate cancer it's a minority platinum sensitive disease.
Of course, and in that study they picked out the platinum responders, but still there's this concept of BRCA-ness that we haven't yet defined genomically. Do you think that's something that may be coming down the pike in prostate? Or their target genes? Are we thinking about it?
Yeah, you need a functional assay. That's the key thing that's missing is some sort of functional cellular assay that could tell us if patients are actually able to repair DNA as opposed to just sequencing the genes and presuming that there's that link. So I don't know the answer to that question. It's a great question.